In last or in previous class, what else we have started or what else we have studied? Still possible in the sense they can mix the chapter. The chapter we have studied. Last class we have started. In last class we have started the chapter microbes in human welfare. Microbes in human welfare. Still past so far we have studied about what are the different kinds of microbes, where they used to grow, and what is the habitat of these microbes, the examples for the microbes, and then we were studying about microbes in household products. Microbes in household product. And in that particular point, we will study 3 bacteria like lactobacillus, that is we are going to call as a lactic acid bacteria. And after that, we will study about the Saccharomyces cerevisiae. And last we have studied the bacteria named as Propionibacterium charminium. So these 3 microbes which were used for the preparation of different kinds of food, and that's what we were mentioned those in a particular point called as microbes in household product. Now we are going to study the microbes in industrial product. We are going to study the microbes in industrial product. Still possible we have studied about microbes in household product. Household product in the sense conversion of milk into curd. We have studied about the preparation of dosa and idli. That is what we have called dough. They were going to use the bacteria. The name of the bacteria is Saccharomyces cerevisiae. Right? Then some bacteria are used for the preparation of some traditional drink. All these things we were discussed in a previous class under the concept microbes in household product. Now today we are going to study microbes in industrial product. So let us start with the microbes in industrial product. Industrial product in the sense, even in industry, microbes are used to synthesize a number of products. Even in industry, the microbes are used for the production of number of products or synthesis of number of products that can those are valuable to the human beings. And here I have 2 examples. One is beverages and antibiotics. In the sense, there are many products which can cause in industry by using the microbes. And here we are going to discuss 2 examples. What are those in the sense? One is beverages, another one is antibiotics. And about beverages and antibiotics we are going to study in this particular class in detail. So let us start with the first one is beverages. Before we are going to go for the beverages, we are going to study one definition. The definition is fermenters. Fermenters are nothing but the large vessels. For example, simple example ಕೊಡಬೇಕಾದ್ರೆ ನಾವೇನು ಮಾಡ್ತೀವಿ, ಈಗ ಒಂದು ಸ್ಮಾಲ್ ಸ್ಕೇಲ್ ನಾಗ ಮನೆಯಲ್ಲಿ ಊಟ ಮಾಡೋದು ಕೂಡ ನಾವೇನು ಮಾಡ್ತೀವಿ, 4 members ಅಥವಾ 5 members ಇದ್ದಾಗ we used to use the small cookers. But when you are going to go for the hotels, the large hotels, that time they used to prepare large quantity of rice. That time they are not going to use small cookers. They used to use the big vessels. Likewise, here in some industries, they used to use the large vessels for the growing microbes. In the sense, in a small scale industry, the quantity ಕಡಿಮೆ ಇದ್ದಾಗ ನಾವು ಮಾಡ್ತೀವಿ, ಒಂದು ಸ್ಮಾಲ್ ಒಂದು ಬೋಗಾನಿ ಒಂದು ಸ್ಮಾಲ್ ಪಾತ್ರೆಯಲ್ಲಿ ನಾವು ಮೈಕ್ರೋಬ್ಸ್ ಬೆಳೆಸಬಹುದು. ಆದರೆ ಲಾರ್ಜ್ ಸ್ಕೇಲ್ ಇಂಡಸ್ಟ್ರಿಯಲ್ ಸ್ಕೇಲ್ ತಂದಾಗ ದೊಡ್ಡ ಇಂಡಸ್ಟ್ರಿದಾಗ ಬಹಳಷ್ಟು ಸ್ಕೇಲ್ ನಾವು ಮೈಕ್ರೋಬ್ಸ್ ಬೆಳೆಸಬೇಕಾದ್ರೆ ಏನಾಗ್ತೈತಿ, ದೊಡ್ಡ ವೆಸಲ್ಸ್ ಬರ್ತಾವು. ಆ ದೊಡ್ಡ ವೆಸಲ್ಸ್ ಅದನ್ನ ಫರ್ಮೆಂಟರ್ಸ್. ಆಸ್ ಪಾಸಿಬಲ್ ನೀವು ಅದನ್ನ ಏನಪ್ಪ ಅಂತ ಡೆಫಿನೇಷನ್ ಬರೀಬಹುದು. And this is very important. One marks question. So when I ask for the final exam, one marks question ಕೇಳ್ತಾರೆ. What are fermenters? Fermenters are nothing but fermenters are very large vessels are required for a growing microbes for the production on an industrial scale. For what? Production on an industrial scale. Microbes are grown in a very large vessels for the production on an industrial scale, and such large vessels are called as a fermenters. And such large vessels are called as a fermenters. Now, these fermenters are used for the preparation of beverages. Beverages is a fermenters. These fermenters you can see even in what we are going to call it as a sugar factories ಹೋದಾಗ ಮೊಲಾಸಸ್ sugar factory ನಾಗ beverages ತಯಾರು ಮಾಡ್ತಾರೆ. ಅಲ್ಲಿ ದೊಡ್ಡ large fermenters ಕಾಣ್ಬೋದು. Okay, so this is the definition or what are fermenters ಅಂತ ಕೇಳಿದಾಗ very large vessels are required growing microbes for the production on an industrial scale are called as the fermenters. Fermenters are nothing but very large vessels which are used to grow the microbes for the production of an industrial scale. Large scale ದಾಗ ನಾವು ಪ್ರೊಡಕ್ಷನ್ ಮಾಡಕ ಮೈಕ್ರೋಬ್ಸ್ ಎಷ್ಟೋ ಬೆಳೆಸ್ತೀವಿ, ದೊಡ್ಡ ವೆಸಲ್ಸ್ ನ ಬೆಳೆಸ್ತೀವಿ ಅಥವಾ ಗ್ರೋ ಮಾಡ್ತೀವಿ. ಆ ವೆಸಲ್ಸ್ ಏನು ಕರೀತಾರೆ? ಫರ್ಮೆಂಟರ್ಸ್ ಅಂತ ಕರೀತಾರೆ. ದಟ್ ಇಸ್ ವಾಟ್ fermenter. This is the first thing we need to know. Now we are going to start knowing about the fermented beverages. How the fermented beverages? Yes, see, in fermented beverages, microbes, especially yeast, we are going to use here microbes, especially yeast is used. Especially yeast have been used for the preparation of beverages. Especially yeasts are used for the preparation of beverages. Here see, microbes, especially yeast, used for the production of beverages. Like what are the beverages are going to produce in a large scale by using the fermenters? One is wine, beer, whiskey, brandy, or rum. These all are the types of beverages which are prepared or which are produced or which are made by using the microbes, especially yeasts. Especially what I told you, yeasts. Especially yeast. Okay, and here whatever the yeast or what we are going to call that as the same yeast which is going to use the name, that is what Saccharomyces cerevisiae. Where you are using this Saccharomyces cerevisiae in previous class or in previous video we were studying regarding microbes in what household product there for the preparation of idli and dosa. And whatever we are going to form the pores by the production of carbon dioxide, that is with the help of what? Saccharomyces cerevisiae. The same is used for the preparation of these beverages. Understood? So allu kela next name the microbe which is used for the preparation of idli and dosa, and name the microbe which is used for the preparation of beverages of Saccharomyces cerevisiae. And this beverage, it is used for the formation of breads also. Okay, in a sense bakery for the preparation of breads also. So this Saccharomyces cerevisiae, it is commonly known as Beverages yeast. BREWERS, Beverages yeast. And this is also going to ask you for the one marks question. Name the Beverages yeast. Beverages yeast is nothing but Saccharomyces cerevisiae. Saccharomyces cerevisiae. I hope this is clear for you people, right? And you are familiar with the Saccharomyces cerevisiae. This Saccharomyces cerevisiae is used for the preparation of beverages. Preparation of beverages. Beverages are nothing but wine, beer, whiskey, brandy, or rum. And after this, what we are going to concentrate here in the sense, depending upon the raw material, depending upon the type of raw material used for the fermentation and the type of different, what we are going to call it as a different kinds of alcoholic drinks, whatever means which kind of raw material we are going to use for the preparation of beverages and which kind of alcohol we are going to produce. Based on that, there are two kinds of beverages. One is distilled with distilled beverages and without distilled beverages. And here already I written wine, beer, whiskey, brandy and rum. These all are the beverages. And here apart from these, in the sense all of these are the beverages. From these beverages, wine and beer, these are produced without distillation. These are produced without distillation. Whiskey, brandy and rum are produced with distillation. And here also, the fellow is going to ask in the sense, the question is going to arise in such a way that name the beverages which are prepared with distillation. That time we are going to write whiskey, brandy and rum. Name the beverages which are produced without distillation. Those are wine and beer. This much you are going to remember regarding what we are going to call about as a microbes in industrial product. First example is beverages. In beverages, you are going to remember fermenters. After that, fermented beverages are nothing but wine, beer, whiskey, brandy, rum. Okay. In this, wine and beer which are going to produce without distillation, and whiskey, brandy and rum are going to produce with distillation. These are the things you are going to remember. And here, these are the microbe which is used for the preparation of different kinds of beverages. That is what Saccharomyces cerevisiae. Okay. How these are going to ferment? Okay. Here see, fermenting this Saccharomyces cerevisiae is used for the fermenting malted cereals and fruit juices. Malted cereals and fruit juices ಅಂದಾಗ cereals are nothing but ಇಲ್ಲಿ ನಾವು ಏನಪ್ಪಾ ಅಂತಂದ್ರೆ ನೀವು ಜೋಳ ರಾಗಿ ಅಂತ ಕರೀತಿರಲ್ಲ cereals ಅವನ್ನೆಲ್ಲ ಏನು ಮಾಲ್ಟೆಡ್ ಅಂತಂದಾಗ ಅವನ್ನ ಒಂದು ನೀರಿನಾಗ ಹಾಕಿ ನಾವು ಬಹಳಷ್ಟು ದಿವಸ ಇಡಬಹುದು ಅಥವಾ ಮೋಲ್ಡ್ ಬರೋ ಮಾಡೋದು. ಅನಂತರ ಫ್ರೂಟ್ ಜ್ಯೂಸಸ್ ಬಹಳಷ್ಟು ಫ್ರೂಟ್ ನ ಜ್ಯೂಸಸ್ ಏನಿರ್ತಲ್ಲ ಅಲ್ಲಿ ಏನಪ್ಪಾ ಅಂತ ಫರ್ಮೆಂಟೇಷನ್ ಮಾಡೋದು. And I told you already fermentation ಅಂದ್ರೆ ಏನು, what is the process or what are the steps involved in a fermentation and what is the end product of fermentation. For that I told you about respiration. Last year the respiration chapter ತೆಗೆದ್ರೆ I know. Okay, anaerobic respiration ಬರುತ್ತೆ fermentation. So you go to that. That is what this Saccharomyces cerevisiae. It is helpful for fermenting malted cereals and food to produce ethanol. To produce ethanol in the sense it's going to produce the alcohol at the end. So this is about what? Beverages. After the beverages, we are going to study antibiotics, which are also means that is also the example which is come under the microbes in industrial products. Microbes in industrial product. So here you all know about the antibiotics, or I can say you all are familiar with the name antibiotics. So many times you have heard about the name antibiotics. Then what is the meaning of this anti and bio? Anti and bio. This is the anti. It is came from the Greek word anti in the sense against. Bio in the sense life. Against life. Antibiotic in the sense against life. And here one point you are used to remember that is very important. This against life in the sense for disease causing organism. Against life for disease causing organisms. Okay, and not this is not for against the humans. Understood? So anti it is came from the Greek word anti in the sense against, bio in the sense life. Okay, antibiotics are against the life of pathogens or the microorganisms which are going to cause the diseases, not for human being. In the sense for the human beings, these antibiotics are pro-life. Pro-life in the sense those are helpful for improving the health status. Understood? So this is very important. Now I am going to start with the antibiotics. I think these antibiotics are nothing but these are the chemicals or these are the chemical substances which are produced by some microbes and kill the other microbes which are disease causing ones, or which are going to cause the diseases. Those are called as antibiotics. Now, what is the first antibiotic or what is the first means antibiotic is developed. The name is penicillin. Penicillin is the first antibiotic which is developed by the Alexander, not developed by the Alexander Fleming. This is the accidental discovery. This is the accidental discovery about the penicillin when the Alexander Fleming is working with the Streptococci bacteria. STAPHY-LOCOCC Streptococcus bacteria. In a sense, for example, this is what we are going to call as this is a petri dish. A petri dish na ಏನಂತಪ್ಪ ಅಂತಂದಾಗ Alexander Fleming ಎತ್ತರ ಜೊತೆ ವರ್ಕ್ ಮಾಡ್ತಿದ್ದ on which he is working ಅಂತಂದಾಗ Streptococcus bacteria. So here suppose this is a Streptococcus bacteria work ಮಾಡ್ತಾ ಇದ್ದ. Unfortunately ಏನಾಯ್ತಪ್ಪ ಅಂತಂದಾಗ unwashed, what we are going to call that one as a unwashed petri dish. petri dish ವರ್ಕ್ ಮಾಡೋ ನಮ್ಮ ಕ್ಲಾಸ್ unwashed ವಾಶ್ ಮಾಡೋ petri dish ನ ನಾವು ಏನು ಮಾಡಿದ್ವಿ ಅಂತಂದಾಗ he found some molds ಅಂತಂದಾಗ molds ಅಂತ ನಿಮಗೆ ಗೊತ್ತಿರಬಹುದು. ಏನಾದ್ರು ನಾವು if we are going to prepare some statues and all and ನಾವು molding ಮಾಡ್ಕೊತೇವೆ ಹೆಂಗ್ ಬೇಕಾದ್ರೆ mold ಮಾಡೋದು. mold ಹೆಂಗ್ ಅಂತಂದಾಗ unwashed petri dish ನ ಏನಾಗಿತ್ತು, some molds used to grow and surrounding this mold he couldn't find Streptococci bacteria. Suppose ಒಂದು petri dish ಇದೆ, ಅದು unwashed ಐತಿ, ಅದರ Streptococcus bacteria ಅದಾವ, ಅದು ನಾವು ವಾಶ್ ಮಾಡಿರಲ್ಲ. ಅಲ್ಲಿ ಏನಪ್ಪ ಅಂತಂದಾಗ ಒಂದು ಮೋಲ್ಡ್, ಒಂದು ಬ್ಯಾರೆನ ಏನಪ್ಪ ಅಂತಂದಾಗ ಒಂದು ಬೇರೆ ಏನಪ್ಪ ಅಂತಂದಾಗ ಆರ್ಗಾನಿಸಮ್ ಅಲ್ಲಿ ಬೆಳೆಯಾಕಿತ್ತು. ಆ ಬೆಳೆದಾಗ ಏನಾಯ್ತು, ಆ ಮೋಲ್ಡ್ ಸುತ್ತಮುತ್ತ ಏನು ಬೆಳೆದಿರೋದನ್ನಾಗ Streptococci ಬ್ಯಾಕ್ಟೀರಿಯಾ ಬೆಳೆದಿಲ್ಲ. ಅದ್ರ ಮೋಲ್ಡ್ ಸುತ್ತಮುತ್ತ Streptococcus ಏನಿರಲಿಲ್ಲ, Streptococci ಬ್ಯಾಕ್ಟೀರಿಯಾ ಇರಲಿಲ್ಲ. That time what he observed in the sense Alexander Fleming, that mold can produce some chemicals. And that chemical it is against the growth of Streptococci bacteria. So that chemical he named as a penicillium notatum. ಆಗ ಅದಕ್ಕೆ ಹೆಸರು ಕೊಟ್ಟ penicillium notatum. ಅರ್ಥ ಆಯ್ತು ನಿಮಗೆ? penicillin, it is the first antibiotics. Okay. What we are going to call that, it is accidentally discovered. When discovered by in the sense Alexander Fleming, when he is working with the Streptococci bacteria in unwashed petri dish, he found some growth of mold, and surrounding that mold he couldn't find any Streptococcus bacteria. In the sense he observed that mold produced some chemicals. That what I told you, mold produce some chemicals in the plant that could not, what we are going to call it as a, that could not able to grow that Streptococcus bacteria. In the sense, what is going to happen here? Unwashed culture plate around which, whatever the unwashed culture plate is there, in which the mold is grown, and around the which what we observed in the sense Streptococci could not grow. And that is due to some chemicals which is secreted by the mold, and he named that chemical is penicillium notatum. And then after what we are going to call that as a, however, it is the full potential as an effective antibiotics. That afterwards the full potential, what we are going to call that one as a antibiotics, was established much later by Ernst Chain and Howard Florey. They are worked on it, and much later, after hence the penicillium notatum, it is what we are going to call as a produced as a potential antibiotics, and that antibiotics is used for the treating American soldiers wounded in the World War II. And for this, what we are going to call that as a discovery. For this discovery, Fleming After this, chain and fluorine, all three together, they are got the Nobel Prize in 1945. They are got the Nobel Prize for this discovery, penicillin discovery in 1945. 1945. After this penicillin, there are many antibiotics were developed. Like for the disease plague, there is antibiotic. Whooping cough means cough in the sense khali kasi. Diphtheria, gall, gutta, and leprosy. Leprosy in the sense ಕುಷ್ಠ ರೋಗ ಇವಕ್ಕೆಲ್ಲ ಏನಪ್ಪ ಅಂದಾಗ ಇದೊಂದು ಪೆನ್ಸಿಲಿನ್ ಮಾರ್ಕ್ ನಂತರ there are the discoveries which we are going to call them as a antibiotics. ಅಂತ ಮುಂದೆ ಏನಪ್ಪ ಅಂದ್ರೆ ಈ ರೀತಿ antibiotics ದ ಡೆವಲಪ್ಮೆಂಟ್ ಆಗ್ಬೋದು. ಆನಂತರ today we cannot imagine a world without antibiotics. Today we are not going to imagine the world without the antibiotics. In such a way that the microbes are got their own importance in what? Industrial products. So this is the second point regarding microbes in human welfare. First point we were studied regarding the microbes in household product, and today we are studying about the microbes in industrial product. In this microbes in industrial product, we will discuss two examples. What are those in the sense? One is beverages, another one is antibiotics. Here beverages we studied the fermenters, then fermented beverages, wine, beer, whiskey, brandy, rum, and here we are distributing means what categorize them like without distillation and with distillation. Then we have studied about the antibiotics. So this is about what the microbes in what we are going to call that as a microbes in industrial products. So I am going to stop here, and in next class I am going to start with the next point which is come under the microbes in human welfare, that is chemicals, enzymes and other bioactive molecules. And next we are going to study microbes in sewage treatment. Okay, I'll stop here. Thank you. I'll continue next.